Welcome to the Environmental Extremes Global Engagement Series presented by the Thermal Economics Lab from Sydney. This is episode 2 and it's the first part. My name is George Havenith and I'll be presenting or discussing this later on together with Caroline Smith from Appalachian State. The theme for this for the 2 papers that I've selected is methodologies. One paper is related to a work on sweat rate and measuring local sweat rates across the body. And the second paper is related to the work I did for my PhD on individual differences. So I'm going to discuss 2 papers in 2 sections. And the first paper I'm going to talk about is a paper which was done by Caroline Smith, at the time my PhD student, and myself on body mapping of sweating patterns in male athletes in mild exercise-induced hypothermia. So what I'm going to talk about is why did we do this, how did we do it, and what were the main outcomes. And the detail is probably up to you to read in the paper and come back with questions to us later on. So why do we need to measure sweat distribution across the body? Well, there's interest from various sides, and what you see here is a thermal mannequin. And these thermal mannequins are in essence heated dummies which simulate the surface temperature of a human. And the more modern versions are also able to sweat. And the sweat is distributed typically over something like 250 sweat glands and then spread out by the skin that you can see here, the skin which spreads the water, so wicks very well. And that means that in the design of it, because most of these have 20 to 34 individual zones, you would like to have a different setting of sweat rate for the different zones. So in order to set that in a natural way, rather than have the same sweat rate all over, you need some information on sweat distribution. Secondly, a lot of modeling of temperature regulation has happened since many, many years, starting probably with Stolwijk with his model, which was used to look at astronauts going into space. And more recent models like the Fiala model have been looking at also a distribution of heat transfer over different body parts. And again, if you want to look at that specific interaction in a specific body part, you want to know what the sweat rate locally is. And once you have that, you can use that in, for example, here we have a CFD model where you see that airstream goes across the body in different areas, and it will affect the evaporation in different areas differently. So knowing what the sweat production is, you can then combine with the sweat evaporation based on the climate and the airflow. The third main application is in clothing design. So a lot of companies are interested in evidence-based clothing design guidelines. So they want to know what should they do in terms of selection of fabrics. And so this is a picture taken from Margherita Cuglia's paper in 2019, looking at moisture absorption by 2 different clothing types, both t-shirts but different materials, across a 50-minute run. And if or when manufacturers know how sweat is distributed, they can make selections. For example, like where do we put ventilation openings, where do we put highly absorbing fabrics, where do we put fabrics which absorb very little but might have other beneficial properties instead. So then the how, and the how starts really with historical research. So we want to know the distribution over the whole body. And Kuno, and I really advise you to try to get a copy of his book from 1956. He's done a lot of work in, I think, 30 years before that even, on sweat. And it's a really good place to start reading about sweat production, sweat glands. And he used a method which is called the colorimetric method. So what he did is he painted people with an iodine starch solution, and that's white. But as soon as you start to sweat and the sweat interacts with the iodine and the starch, it turns black. And that's what you see in this picture. So that's a great way of finding out where people start to sweat and which areas have sweat, but you can't have any quantitative judgments. So we want to quantify the amount of sweat, so we need a different method. And for that, the obvious method, which is shown here in these pictures, which were currently given to me by Cristiano Mercado Moreira, who worked in Nigel Taylor's lab, is to work with ventilated sweat capsules. So with the sweat capsules, you blow in dry air into the capsule, you extract the air from the capsule, which has absorbed the sweat, and you analyze the air going in and out for moisture content, and that gives you a sweat rate. So downside of this is very small areas. So you see that these are quite small, so 3 to 9 square centimeters. Typically they cover, even in these high density experiments, less than 2% of the area you're interested in. The good part is, of course, you have a continuous measurement. One consideration, which is interesting for you maybe to discuss compared to the other methods that we use, is this blows in very dry air, so it keeps the skin dry, which may have an impact on promoting sweating and maybe slightly skew the results. And you will later see that the other method that we use has different issues, potential issues to look at. One other downside we had in our lab in the last couple of years, that we had a couple of people showing a reaction to the collodion glue, which doesn't seem to be that common, but we had 2 people show it, so that makes it more difficult. And finally, if you look, for example, at the top left picture of the foot, with that foot you can of course not run. So that's another thing which we want to do, and therefore we had to look for a different method. So we wanted to cover as much of the body as possible, and therefore we had a schematic like this, where you see all these different body parts which would have absorbent patches that absorb the sweat put on them. And these are all anthropometrically sized to each individual person. So the red lines here are patches that we had there, but we then threw away because, for example, the top one in the neck avoids any sweat coming from the face or the neck to go into the patches just below it. And the armpit patches we also didn't use. So just to show you the principle of this, so what you see here is one of our students at the time, this was for the experiment we did in 2008, having prepared the cutouts for the different body parts, and she's now placing this on a plastic sheet, and that's what we need for fast application, as you'll see in a minute. So all the patches are sized by individual anthropometrics. We would only cover a third of the body at one time, in order to not to overheat the person if they can't evaporate in that period. So we need short measurement periods, and we need to avoid saturation and moisture buildup. So where the sweat capsule has very dry air, the risk with covering the skin is, of course, that you get moisture build up and potentially hydromeiosis occurring, so reduction of sweat rate due to the application. In this case, given that we only applied it for 5 minutes and that we only use less than 5% of the absorption capacity of the technical absorbent, this should not be an issue. It's important, of course, when you're finished, as you'll see in the next slide, that you put all the patches in a plastic bag very quickly to avoid mass loss while you're doing the rest of the procedure, and you can then weigh them afterwards. And again, the technical absorbent has the advantage that once it absorbs the moisture, it doesn't really release it very well, and that's quite different if you would use just plain cotton, for example. So fortunately, also later after this study, there was some validation in comparing this to sweat capsule, and that's a good read from Nathan Morris, who published that in 2013 in JAP. And he compared sweat patches with the ventilated capsule, and he found really good relations between the two data. So just to show you the experiment, so we have a person who has been running for half an hour. So this is again pictures from 2008, but the principle is the same. So the person was running for half an hour, person comes off the treadmill, is towel dried very quickly, and then as you see, the sheet with all the absorbents is placed on the body. And then a stretch shirt is put over it, or stretch trousers, or whatever section you're looking at. And then the person goes back to running for 5 minutes. And in those 5 minutes, it absorbs the sweat coming out of the sweat gland. And after that 5 minutes, the person is undressed, the patches are all put in plastic bags and weighed. So to show you the protocol, 1 hour run total, 30 minutes at 55% VO2 max, and then 30 minutes at 75% VO2 max. Sampling only took place in the last 5 minutes of each period. So most of the time they were just running in normal clothing. It was 9 fit males in this case. And on the right-hand side, just as an illustration, you see the gross sweat loss data for the experiment in relation to the metabolic rate. And interesting to see, and of course, if you look later on with experiments done, especially in Ollie's group, but also others, you see a very strong relation here between sweat rate, sweat production, and metabolic rate. But interesting is that the lines for intensity 1 and intensity 2 are not continuing from each other, but actually there is a shift in the line upwards for the individuals. So let's look at some results. So this is the result for the low workload, and I'll put straight away one up for the high workload. And you see there's quite a big variation in distribution with sweat rates from the forehead at the high work rate around 1700 grams per hour, in the central back, so the spine, around 1200 grams per hour, going all the way down to places like fingers, 144 grams per hour. So a huge variation in sweat rate across the body. So for most people, for the first group, we did the whole body, and the head group was partially a different group because obviously not everybody wants to shave their head, but we corrected sweat rates based on the people that had done both experiments, and we related those to each other. So what were the main findings? So the first thing here is, if you look at the torso, you see that sweat decreases from medial to lateral. And at the scalp, this is actually the opposite way. It increases from medial to lateral. Then on the extremities, so upper arm to lower arm and upper leg to lower arm, we see an increase from proximal to distal. So if you look, interestingly, the shoulder have a high sweat rate, then the upper arm a low, and then the lower arm, again, a higher sweat rate. Hands and feet have low sweat. If you compare the two intensities, you would see that all areas increase from intensity 1 to intensity 2, except for the feet, which didn't have a significant increase. And that's information that was later confirmed by Anna West in her recent paper on foot sweating. So if we look at the results, what can people do them? If, for example, so the first thing you can do in terms of the mannequins, you can copy these values of sweating to the mannequin settings in the program that controls the mannequin. If we think about the models, you can use these indeed to look at sweat production, therefore cooling power in different body parts. And for clothing design, just examples here, these were some examples which were done straight after this paper came out, where the spine was the area around the spine was designed with extra ventilation opening to make sure that that extra moisture that's on the spine evaporates. So this is normalized. So what we did is we added up all the sweat rates across the body and calculated the average sweat rate overall, and then for each section we divided the value by that average. And that means it's a bit easier to see in terms of the colors, and it also makes it easier to compare different levels of sweating. So in the previous slides, you could clearly see that we had a higher sweat rate at intensity 2 than intensity 1. But if you look at the distribution, you see that the main items of the distribution are visible on the left as well as on the right-hand side. So the distribution changes a little bit when you start to sweat more, but not as much as we actually thought beforehand. Two other points to mention: we did not find any correlation of local sweat rates to local skin temperatures, which was important to see whether putting on a patch which would warm up the skin a little bit would affect the sweat rate. And finally, no relation of local sweat rate to the distribution of sweat gland densities. So if you look into the literature, you'll find that the distribution of sweat gland density does not match what you see here in sweat production. And that implies that it's more about the maximum amount of sweat pushed out by each individual sweat gland, that differing across different areas than it's the number of glands determining this. So work for the future, or work that has followed up since this. Firstly, Caroline did some work on females to compare to males. And then keep going on from that, Lee Arlegui at the moment is doing work on children's sweat rates, and Nicole Kuhl has done work on sweat rates in older people above 65 years and comparing that with younger people. So the age is an important aspect. Sex differences are important aspects. And Caroline did a paper published in 2019 in which she looked at acclimatization changes in sweat distribution in the upper body. So one question I get asked very often is, why is the distribution like this? And it's an important question because in some ways it's not really logical, because the back sweating so much more than the front is not necessarily beneficial for heat loss. When you're running or any movement, the front is exposed to much more airflow than the back, so it will evaporate more moisture in the same temperature, and therefore it's very likely that the back and the spine are over sweating, so there would be unnecessary sweat production there, which doesn't lead to extra cooling. The interesting question is there an evolutionary aspect to that? And unfortunately, we can only speculate on that. And there was a suggestion made to me by an evolutionary biologist at some point, whether it would have something to do with the development of this pattern before we or ancestors became bipedal. But that's a long jump to make. Okay, that's the first paper, and I'll do a presentation on the second paper shortly. Thank you.